Hello, folks. Welcome to the next session. My name's Alasdair Coles, and I'm from Cambridge.
And I'm Omar Abdel-Mannan from UCL.
So Masood asked us to be subversive, and we have chosen to be subversive in this session by presenting you with three different facets to neuroimmunology that you might well not have thought about. So first up is David Hunt from Edinburgh, who is going to tell us about a form of neuroinflammation that has nothing to do with multiple sclerosis. So this is all about interferons and viruses in the brain. David, thank you very much.
Thanks very much, Alasdair. And firstly, this has been a really brilliant day. I've really enjoyed it. And I also wanted to thank you for calling the session Neuroinflammation, because I'm going to talk a lot about innate immunity. People often ask, well, what do you mean by neuroinflammation? How is that different from neuroimmunology? Because neuroimmunology just sounds a little bit cooler. So I'm going to show you today that innate immunity is also important in human neurological disease. So I thought the best way to start with that is probably with a case. Like all good cases, this started with a neurosurgical referral, but the history went back a long time. So this is a patient who has had accumulated autoimmune or immunological diseases, at least 3 of them, over the last few decades. Ankylosing spondylitis, he had large joint arthritis as a child, and then was developing recurrent fevers and a low-grade lymphocytic meningitis. And there were concerns about the meningitis. He was actually under the neurosurgeons because of raised pressure. And the question was whether or not this was all a chronic meningitis driven by infection because of his immune treatments that he was having. He's had multiple immunotherapies at that point. So the question was whether it was an opportunistic infection. But actually, going back, he had had raised inflammatory markers for decades. He had very abnormal discs, and he had this persistent raised CSF lymphocytosis. He was losing vision very rapidly. And so this gentleman didn't have an infection. He had a mimic of infection. He had a gain-of-function mutation in his NLRP3 inflammasome, which is involved in sensing patterns and activating the immune system. So that's particularly important because this only responds partially to other immunotherapies, but responds extremely well because the inflammasome links through and causes IL-1 beta, so it causes a cytokine production, which can be blocked with receptor blockers such as anakinra or canakinumab. So this gentleman, having had ESRs, CRPs for decades in the hundreds, within weeks, when all of the antibiotics were stopped, all of the treatments that he was on was stopped, was brought under full control. All of his multi-system inflammatory disease ceased. So it's a very good example of how thinking about innate immune activation, particularly on a genetic level, this is the sort of thing that usually presents to pediatricians, but can occur late in life and can help explain polyautoimmune disease. So the rheumatologists are much more familiar with this, and they have a much better sort of understanding of how we characterize autoinflammatory states, typically these conditions such as CAPS and the Mediterranean fever, which are caused by innate immune activation with exemplars of monogenic disease. And then that moves over to a spectrum where you get much more autoimmune, characterized maybe by autoantibodies and things which involve memory and forms of autoimmunity. But this spectrum also exists in neurology, and I'm going to talk a little bit more about how innate immune activation unchecked can cause devastating neurological disease and how it does that. So this is maybe something that we'll all be familiar with, which is cell-intrinsic innate immunity, how we can respond rapidly in a non-specific way to invading pathogens by recognizing motifs such as nucleic acids as non-self and being able to activate antiviral cytokines such as the type 1 interferon system. There's a long history of the type 1 interferons. This is Alex Isaacs, a Scottish scientist who, together with John Lindemann, discovered the interfering substance, the antiviral substance, in 1957. It took a long time for that to be believed because it was so species-specific. This work was done initially in chicks. But the type 1 interferon system has been very clearly and well elucidated over the 6 or 7 decades following that. And typically, the pattern, the motifs that are recognized in nucleic acids of lots of different flavors, and those are recognized either by TLR sensors or by an increasing array of cytoplasmic nucleic acid sensors. Most recently, the newer kid on the block is a CGAM sting pathway. And that leads to activation of the type 1 and production of type 1 interferons, which then undergoes downstream signaling and activation of antiviral mechanisms. So why is that relevant to the brain? Well, this is really an important system for protecting the brain against viral infection. If that system doesn't work, then people are predisposed to things like herpes simplex encephalitis. So STAT1 deficiency was first described in about 2003, 2004, predisposing to herpes simplex encephalitis. But also, if you have a trigger-happy system, so if this is too easily, if you have too many of these nucleic acids being produced, then this can lead to, or you have gain-of-function mutations in this pathway, this can lead to a trigger-happy overexpressing system where damage can occur because of sterile inflammation. So that's typically considered, you have almost an extreme phenotype here, which is a host defense deficit of interest largely to infectious disease physicians. But also here, you have an extreme phenotype of monogenic disease with an overactive interferon response. But we see in many common systemic autoimmune conditions overactivity of the type 1 interferon system as well. So this is a child that was born with what was thought to be a congenital viral infection. It was recognized in the 1980s that a subset of children who were thought to be born with congenital infection but no organism were found actually had a Mendelian mimic of congenital viral brain infection. So typically they would be negative for all of the organisms that could cause this, but they would have raised CSF interferon alpha, raised systemic interferon alpha, but they would often sustain severe brain disease. And these are often caused by, their recessive conditions often caused by biallelic mutations, which cause loss of function in nucleases, which degrade nucleic acids, which then lead to a buildup of nucleic acids within cells, which can then stimulate these nucleic acid sensors and thus stimulate interferon production. We also see this in systemic autoimmune disease, classically with a lupus. So we see in about 80% of people with lupus have elevated type 1 interferon signatures. And also it's thought that rare variants in these type 1 interferonopathy or Aicardi-Goutieres syndrome genes can confer risk for lupus. So my lab has been looking both upstream and downstream to try and identify what the potential mechanisms of this are. I'm going to talk purely about downstream today because I think that this is probably closer to clinical translation and of relevance. But a lot of work studying upstream is trying to work out exactly what the mutations are and how they activate these pathways. But it is downstream that we're going to focus on today. So one of the problems, one of the difficulties that has beset the field is that interferon alpha in particular is an extremely potent cytokine. It's a very low abundance protein and has classically evaded detection by ELISA for many years. So many people, I mean, I first learned ELISAs in Alasdair's lab. Things like interferon gamma, it's relatively straightforward to have an ELISA. But actually, you need to have a special assay, really, to measure interferon alpha directly, rather than indirectly, which has been used for a while. So we've been doing a lot of patient-centered studies to try and identify where this neurotoxic interferon is coming from, but then using modeling approaches to try and work out how it is having its deleterious effects in causing brain disease, with a view to trying and understanding the condition better, developing biomarkers and working towards clinical trials. So to do this, this is work from the Crick, Adrian Hayday and Kai Sand, who have isolated antibodies from patients with air deficiency. So this is actually a classic monogenic autoimmune disease, air being a master regulator within the thymus. These patients develop extremely high affinity antibodies against their own cytokines, and they neutralize their own cytokines, and they develop both autoimmunity but also chronic fungal infections and candidiasis. But they develop extremely high affinity antibodies against interferon alpha. So these antibodies when paired up and used with the Samoa platform, which many of you will be familiar with for the use for neurofilament, is a very sensitive way of being able to detect interferon alpha. So this is work that was done with in collaboration with the Pasteur Institute. So you can see here that the conventional ELISA, this is the lower limit of detection of interferon alpha, and this for the digital ELISA. So it's really 3 orders of magnitude, and this is where all of the action is. Right? So it's really, this assay is really helping us understand a lot more about what interferon is doing. We've measured this across multiple disease cohorts. You can see lupus patients in the middle and patients with Aicardi-Goutieres syndrome, this type 1 interferonopathy at the end with extremely high, it's a log scale, extremely high levels of interferon alpha. The question is, where is this neurotoxic and interferon coming from? Is it coming from the blood? Is it coming from the CSF? So this is a patient who is in my clinic who was presenting with what was thought maybe to be a calcific leukodystrophy, but he also had a brother who died at birth of a brain infection. And same mutations can cause very different phenotypes in patients. And he had a very strongly elevated blood and CSF interferon alpha. And he had biallelic mutations in ADAR1, which cause AGS. So we're trying to work out whether the neurotoxic interferon is coming from the CSF or coming from the blood. So we've done a lot of paired studies with healthy controls, and MS, which isn't associated with interferon, actually gets better with type 1 interferon, slightly better. But we can see in these patients with cerebral interferonopathies that they have much higher CSF concentrations than they do blood. They do have high blood concentrations, but it's much higher in the CSF. And when we look in the clinic at our patients who have got paired samples with lupus, then we see exactly the opposite. So we see high blood levels, but low CSF levels. So we wanted to try and model and understand the consequences of constitutive high interferon alpha levels in the brain. And to do this, we've been working with colleagues actually during the pandemic at the University of Sydney, who have generated mice which overexpress interferon alpha, and we breed these both between Sydney and in Edinburgh. So this is an astrocyte-driven intracerebral production of interferon alpha. And these mice develop very, very similar pathology to what we see in particularly the children with AGS. You can see here calcium on the CT scan. You can see calcium on the micro-CT here. But the neuropathological findings are very similar. We think this is a good model to try and understand what the targets are of interferon alpha within the brain. There are obviously lots of different cell types. And so which are the cell types that really respond? So we've used 10x single-cell sequencing to look at multiple cell types within the brain of these mice, either wild type or those which overexpress interferon. And the bottom line is only 2 cell types within the brain really seem to care. So quite a lot of cells, and surprisingly, your neurons, your oligodendrocytes, your astrocytes, don't really mount much of a response, and they also don't have the capacity to respond to high levels of interferon. But your endothelial cells and microglial cells both do, and the endothelial cells probably more than microglia. And so the effects of chronic interferon alpha signaling on microvessels is really quite profound. These microvessels undergo very profound transcriptomics shifts. They also they upregulate all kinds of things, cell adhesion molecules. They develop these rather peculiar microaneurysms which come off capillaries. They develop blood-brain barrier leakiness, and they become extremely neurologically sick. And And we can see at a histopathological level a very distinctive interferon-driven microangiopathy, which encompasses many of these changes that we see both in this mouse model, but also in children with AGS. This is actually a patient with a SAMHD1 mutation who, at the age of 10, had an intracerebral hemorrhage, and they also have multiple aneurysms. But we can demonstrate that it really is the endothelial cells that are mediating the disease by floxing out the receptor just on the endothelial cells. So we can create mice that overexpress interferon, but they lack a receptor either on the endothelial cells or on the microglial cells. And it's the ones that lack the receptor on the endothelial cells that actually revert almost to wild type. So these mice develop virtually no brain disease at all. It's showing really that a lot of the secondary neurodegeneration and the change that we see is mediated through the microvasculature. When we do the same on microglial cells, we see no difference whatsoever. So we think that really it is the cerebral microvasculature that is critically vulnerable in these children and some of these adults with cerebral interferonopathies that really mediate the neurotoxicity, the direct neurotoxicity of interferon alpha. That's, I think, an important way for us to understand and conceive the disease, and also when we try to think about where the critical points in the receptors and where they are anatomically for us to target therapeutically. But also with, I don't have many of these patients in my clinic, but I have quite a lot of patients who have lupus in the clinic, and people with lupus develop brain disease as well. And anyone who's worked in that area knows that that is a very tricky, complex, heterogeneous brain disease. We're trying to work out the degree to which actually systemic interferon alpha, so essentially circulating blood interferon alpha rather than CSF interferon alpha, can drive brain disease and be neurotoxic as well. So when we look in patients with lupus, when we first developed and were involved in the development of this assay, we looked in lupus and we saw a very striking effect of interferon concentrations and associations with many aspects of lupus with low complement levels. But also we saw in patients who had very high levels of interferon alpha, we saw both an increase in the number of autoantibodies and the percentage of individuals with autoantibodies. So a real broadening of the autoantibody repertoire in patients with very high interferon levels. And we've been also working, this is Rake Behrend, who I work with at the University of Bonn, in developing a conditional overexpressing mouse that allows us to unequivocally work out the direction of causation, because many people think that it is also possible for high antibody levels to increase interferon. So working out the direction of travel is extremely important. So this is what was called an LSL allele, so it's got a flox stop, so that when you can get a Cre driver from your cell type of interest. So, for example, from dendritic cells using a CLEC9A Cre, it will remove a stop codon and interferon will automatically be produced in the cell and overexpressed in the cell. So you can get cell type overproduction of interferon. And when we do that in these mice, we see that these mice develop anti-nuclear antibodies, and they develop a spectrum of autoantibodies, which we're further characterizing at the moment. The reason we're particularly interested in that is we're interested to work out the degree and the repertoire of brain antibodies that are being produced within that, because we think it's very likely that interferon alpha plays a role in the production of neurotoxic antibodies, and that may well be an important role, important for a subset of patients. So these are 4 patients in my neuromyelitis optica clinic. The first one had systemic mastocytosis and was receiving interferon alpha therapy for that and developed longitudinally extensive transverse myelitis, as did the second patient who was receiving interferon alpha for their hepatitis C. Patient 3 is a young girl who's got an activating IFIH1 mutation, which causes a cardiac UTI syndrome, but actually at the age of 4 developed further weakness and actually had aquaporin 4-mediated transverse myelitis, compounding already the brain disease that she had had. And patient 4 is a patient of mine who has got lupus and multiple autoantibodies. He developed aquaporin 4 associated, and she has, within our cohort of lupus, the highest interferon levels of any. So we think it's likely, and we're currently working out why systemic interferon alpha can also be neurotoxic, but by a different mechanism, not by damaging the microvasculature, but by the production of neurotoxic antibodies. So, just to summarize, interferon alpha is a very important antiviral cytokine, and it can be neurotoxic. But it can be neurotoxic in different ways that's relevant to how we think about how we're going to treat these patients in the future. If the interferon is coming from CNS or brain origin, then it causes a microangiopathy, which can be devastating. And if it is of peripheral blood or peripheral immune origin, it can lead to neurotoxic autoantibody formation. And so we're developing some of this work with some of the work, for example, on interferon alpha quantification for biomarkers within clinical trials, which have been very much needed because of the difficulty of measuring and quantifying interferon historically in the past. So just to summarize, rheumatologists often conceive of immune disease this way, but I think we should be maybe thinking of this as well with some of these monogenic autoinflammatory diseases. They provide insights, I think, into how some of these other autoimmune diseases may arise. In summary, I think that these pathways, as you saw with that very first case, are potentially very highly targetable. So it's just to say thank you very much to my lab. I'm funded by a Wellcome Senior Fellowship and also in the current QQR of the UK Dementia Research Institute. So thank you very much.
Thank you very much, David. And very quickly before, from the floor, if you put your two stories together, can you see any intrathecal autoantibody production in the cerebral interferonopathies?
We haven't looked, but it's a good question. I don't know.
Okay.
We haven't looked.
Work on, please.
Does interferon alpha have any role in neurodegenerative conditions, amyloid, Alzheimer's, those sorts of things? Because you talk about the endothelium being important.
So, I mean, there's an increasing literature on the role of type 1 interferon, I think, in neurodegeneration. It's obviously potentially a target, therapeutic target. It's always quite difficult in that situation to tease out chicken and egg and the degree of contribution of inflammation to degeneration. But I think that there's certainly been, if not knockout experiments with various Alzheimer's models which show that it could be a potential target. So I think that's something that people are looking at.
Great. Thank you very much, David. Thank you.
I'd like to introduce our next speaker. It is Dr. Arman Eshaghi, who is a PI and NIHR Fellow at Queen Square MS Centre and also an affiliate member at the Montreal Neurological Institute. He will be talking today about MS and artificial intelligence, how it's transforming MS care. Thank you, Omar.
Thank you very much. It's a great honor to be here today. I would like to thank the organizing committee for having invited me. The talk is going to be radically different from David's, and I hope it also changes the kind of thinking at the end of the day. These are my conflicts of interest. What I'm going to talk about the next 15 minutes is just giving you an overview of what we call AI or artificial intelligence, where the overall just looking at all the different aspects of potential applications of AI in medicine, not just in neurology or even in multiple sclerosis. I'll give you examples where we have potential for use of AI and also how the future will look like. Half of my talk is going to be about the future because we'll see the evidence base is extremely poor at this point in time. So most of it is really speculation and opinions about the future. So we start from definitions. Artificial intelligence or AI can be any system or any set of technologies whose outputs can resemble human intelligence. And how you define human intelligence, I think, is difficult, and it differs between different schools of thought. So itself is a bit of a vague term. What we usually refer to in scientific literature when we talk about AI is actually not AI, is a subset of AI that is called machine learning. Machine learning is a set of algorithms that can learn input-output relationships. So in the case, if you think of a neurological disorder, that's usually the inputs we can provide to the model, could be imaging, could be any sort of laboratory data, OCT, and so on and so forth. The output could be the prognosis, could be classification, diagnosis. And the bit that is becoming a hot topic these days is part of machine learning that we call deep learning. So these are kind of algorithms that have multiple layers of neurons, artificial neurons that can learn from the data. But what's the promise? The promise, irrespective of the medical field you look at, is to individualize medicine. So my expertise is in multiple sclerosis. We have more than 2.8 million people in the world living with MS, and we treat patients similarly. So what we do at the moment is very much empirical medicine, and the promise of any technology is to move away or move towards individuals, move away from having one size fits all approach. That's the promise. But where are we with this AI or machine learning? When you look at the FDA, or also you can in this country MHRA or EMA, the FDA is the only one that keeps a public database of the AI algorithms approved for clinical use. We see that there is a exponential growth, as you see here. Also in 2023, we have up to July, but also even in 2023, we see that there is almost a doubling of the number of approved algorithms for use in clinical care. When you look at these algorithms already approved, most of them are actually in the field of radiology. And when you look at neurology, we have about 19 or 20 approved algorithms. So it's really a nascent field. Is really early when we looked at how it's evolving. And I also checked last night, so some of these numbers show us exactly where we are now. And where it relates to multiple sclerosis, I think, as we speak, is the area on radiology or neuroradiology. And one of the areas also in neurodegeneration, we have at least 4 or 5 different approved algorithms. But basically what all of them do, they're applied to brain MRI, and they provide you with some sort of report that can tell you, for example, the brain volumes, and can tell you where, for example, a patient might be in their journey, whether it's in neurodegeneration. This is specifically for Alzheimer's disease, not even MS. In MS, you also get hyperintensities or lesions and disease activity over time. But one example of AI, which is the area I work on, is, as I mentioned at the beginning, is to try to use AI to come up with the ways to classify diseases. So this is not specific to MS. It can be done, and I think it's a general idea that you can move towards the biological classification of disorders, especially the disorders that we have been tending to classify based on symptoms. So instead of having, for example, a top-down approach, we could have a bottom-up approach for trying to classify a disease based on the biology. This is the example of ATN, a beta tau neurodegeneration ATN classification that is proposed for use in Alzheimer's disease and similar disorders. And what we see is that these kinds of approaches, they provide you with ample opportunity to use AI, to use large data sets in a way that is unbiased. It doesn't necessarily take into account symptoms, but tries to come up with a biological classification. In MS, as some of you may know, we use symptoms to classify MS. We have patients with relapsed onset MS or relapsing remitting MS, most of whom within 10 to 20 years, they develop a secondary progressive course. And we have also a minority of patients who start with a primary progressive MS. Again, the problem is very similar to what we have in other neurological fields. The types do not necessarily match with the underlying biological mechanisms. And I think this is one of the areas that we see the field moving towards, even using the mechanism-driven frameworks to classify disease, and I think this is the area AI can be very helpful. So this is one of the part of the work we have been doing. We've been trying to use MRI for the classification of MS. This is applied on more than 6,000 MS patients from clinical trials. We've been using the MRI data and MRI modality that are widely available. That includes, for example, brain volumes, as you see here, and also lesion volumes. What you see from the left to right, we have 3 different groups. The question we asked from the AI system, these are kinds of systems called unsupervised systems, meaning that they can look at the data and come up with a way, for example, in this very specific case, how a disease temporally can progress. And the underlying data provided us about 22,000 patient years of data. So that's a huge amount of data we're looking at. What we see is we found 3 different groups of patients. So the 3 was the number that could best explain the temporal evolution of MS. One group that you see on the top, patients which we call lesion-led, patients who have early hyperintensities. Patients who we call them white matter-led patients, patients who have diffuse white matter damage. We use T1, T2 ratio, which is a semi-quantitative way to measure changes in white matter, and patients with cortex-led, patients who have early neurodegeneration, as shown on the changes on the cortex. And we, in phase 3 and phase 2 clinical trials, we looked back in 3 different clinical trials. We saw that patients, what you see in the blue one, the lesion-led patients, they had about 80% increase chance the chance of responding to treatments based on a disability outcome, which we call Expanded Disability Status Scale, or EDSS. So the story here is that you can even have a system that is completely blind to the symptoms, but it can come up with classes that might be helpful, for example, when predicting treatment response. And as you see, for example, patients with cortex-led, they were least responsive to treatments. This was on clinical trials. So where else AI can help to push towards? One area which is the current focus of my research is to pushing these sorts of algorithms from clinical trials, from research into the real world. And one of the most important parts when you want to translate a kind of tool such as AI to the real world is to be able to work with the real world data. And that is where the data is messy, the data is missing. So we're working, and this is already established, that we do have ways, for example, to segmenting a T2-weighted MRI. T2-weighted MRI is a kind of MRI that's not really used for quantitative measures. But we are able now, thanks to the recent advances in computer vision, to be able to segment those and extract the biomarkers that are not usually used. For example, we'll see here we're looking at a phase 3 trial called Oratorio. This is a trial looking at ocrelizumab versus placebo in patients with primary progressive MS patients. We see using T2-weighted MRI that is really not used for this purpose, we can detect the rate of atrophy between different patient groups. So the promise, again, of these methods is to repurpose the existing data we have in the hospitals or hospital archives to ensure that we can have algorithms that are not just for purely academic use, but also for potential future use. What I'm going to talk about the next 5 to 10 minutes, coming towards the end of my talk, is to see where we can go next. As I said, the evidence base for use of AI is extremely poor, specifically in neurology, as we also looked at the number of approved AI algorithms in clinical care. So why is that so? It's because, like, I think and I deeply believe that AI or any sort of algorithm shouldn't be treated differently from treatments, if we want to use that in clinical care. So what you see on the bottom in yellow is how a treatment can come about and to be translated to be used in clinical practice. For example, Alasdair can tell us about alemtuzumab that has taken 50, 60, maybe 70 years from the time that you develop even the molecules towards the running the clinical trials. It's very much similar and should be similar for AI. We have early development phase. We do have a clinical phase where you run algorithms in clinical trials. That has been done, not in neurology, but in a lot of other fields, for example, in cardiology. And usually those are pragmatic trials. Then it goes through a regulatory phase. I showed you a few approved algorithms, and so on and so forth. And it should also, of course, be sustainable through health economics studies, that this can be funded through healthcare systems. So one of the reasons we don't have any AI algorithm for use is because it's a long journey, really. It's very much like having a new treatment. Looking at the literature, we see most of the randomized controlled trials, again, done in other fields. And nothing really in neurology as of now. And also, again, the radiology comes on top. These are the, again, randomized controlled trials. Again, another reason that I think in the next few years we've moved towards using more RCTs in AI and see how that changes. I said a lot about radiology because, as I said, lots of algorithms we use, they are focused on MRI, but of course, as we have seen today, that's MRI is just part of the story, is not really tells us the full story. One of the areas that we see is the ability of AI models. I'm sure you've heard of the ChatGPT and the large language models these days, the ability to understand human language. And what we see on the left is the huge advances that we see, even, for example, answering to USMLE, United States Medical Licensing Examination. These are models that can now really pass those exams. That doesn't mean they are doctors, but the models we have, they understand language, they're able to respond. And what you see on the right is a study published in Nature where for patients with sepsis, the question was, based on the clinical notes, what's the probability of this patient being readmitted to the hospital? And what we see is that both AI algorithms and the clinicians, they had a similar prediction accuracy for whether or not this patient is going to be readmitted post-sepsis. So again, we can argue that there is the possibility of using this in real world. Well, one other area that we are moving towards with new models is the ability to use multiple different sources of data. So we mentioned the imaging. We also looked at the language, but it's possible to mix these two together. There are, again, this is the area that is extremely recent. Lots of the papers are not even peer-reviewed. I'll show here the work done in Google, also partially at Google DeepMind in the UK, is a model called Alasdair. So Alasdair is one of the models that has looked at millions of chest X-rays along with the radiology reports. And usually when you develop an algorithm, you have input and outputs. And these sorts of models that can learn automatically from the language and separately from, for example, imaging chest X-ray in this case. And as you see, the model is able to classify, for example, lung lesions when it's exposed to new chest X-ray. And what is really interesting is that these models have never been told explicitly that, for example, this is a lung lesion or not. They can, if you provide them with a lot of data, they're able to do that. But as I said, if you provide them with a lot of data, there's a big problem when it comes to AI and data. These models are data hungry, and we do have social bias intrinsic to a lot of the data we have. What we see on the left is an AI algorithm used in the electronic healthcare records in California that was used to refer patients based on the notes and refer them from primary, secondary care to tertiary care. And as we see here, that the risk score provided to the patients with African American origin in the US was higher, meaning that you need to be sicker if you're black to be referred to based on the AI. And the reason for that is simply because these models are based on the data we have, and unfortunately, data is biased. So one of the, I think, potential issues is that these sorts of models, once they look at a lot of data, they're going to propagate bias. We have a similar issue in the literature for MS. For example, we see recently we're just completely changing our thinking about how MS affects minority groups. We know that, for example, women with African American origin, they're also similarly likely to have MS. This is exactly the opposite of what we were taught, for example, in medical school. So I'm going to finish my talk here. We looked at the definitions of AI. It's very much a nascent field. There is emerging use. There's a lot of things that are changing even over the past 3-4 months. We saw that there's a lengthy path to translation. There should be realism when it comes to using AI in practice, and I think we need at least another 5 to 10 years to see anything really clinically useful for MS. Thank you for your attention.
Thank you so much, Arman. Do we have any questions for your very interesting talk? Got one here.
Thanks for excuse me. Thanks for an interesting talk. I just thought of one small problem, that when trying to make a correlation, using MRI to classify MS classes, types rather, there is difference in it's hard to correlate between an MS lesion as seen on MRI and the EDSS score because of cerebral reorganization and plasticity. And for example, like the total number of lesions that you see post-mortem on an MS patient it's a lot greater MS lesion, number of MS lesions than would be clinically seen. So how would you teach a computer to understand the difference between looking at the cerebral reorganization part of how much that is translating, a lesion is translating into an EDSS score, if you like?
Well, we cannot be better than our data. So if you can make that decision or that kind of judgment clinically based on the data that's in front of you, that can be embedded within any sort of algorithm. But I agree with you, there's a lot of and that's not necessarily AI's or computers' issue. MRI is not highly specific to lots of things we said. So I think a potential solution would be to use multimodal data that can be realistically acquired in clinical practice. Again, I agree with you, a lot of things relevant to, for example, cortical reorganization could be functional changes, for example, they are not really acquired in practice. But again, if the data is there, or a modality or a source of data that you think can be related to that, could also be, it could be just clinical examination. That can be embedded within with the algorithm. We can use fMRI. The problem with fMRI is not really used in clinical practice. So if I go to a remote village in this country, and we run the same algorithms in hospitals where there are no academic centers of excellence, that wouldn't be useful. So I think this is a very good point. One of the issues we have with AI is the problem with equitability. If you come up with a very fancy multimodal method which is not really applicable across the world in different settings, that is going to cause an issue. But yes, you can incorporate all sorts of advanced measures.
If there's I've got one question, if that's okay. More big picture, what do you think is the biggest barrier at the moment for the applicability of AI in MS randomized controlled trials? Because we're seeing it more in RCT, in real-world data, but not in RCTs.
Yeah, I think it's a lack of really good algorithms or ideas, I have to say. And again, you could say, why don't we run clinical trials for treatments? It's possibly because we don't have a really good treatment that has can provide a high enough treatment effect to then justify follow-on investment. I think a lot of things I showed you, I think goes also back to the previous question. It's really hard to correlate what we have, could be on MRI. Again, the algorithms are not better than your data. With the data we get and the clinical impact. If you can have a minimum, and I think that can be justified by nice guidelines, then that justifies further development.
Great, thank you so much. Go on to the next speaker now.
So when I read a paper, I have various reactions to it. That's cool, that's nice. The highest accolade I can give a paper is, oh, I really wish I'd done that. And I remember that with David's paper in the New England Journal of Medicine on the microangiopathy of beta interferons, and Arman's paper in Nature Communications on the phenotyping of MS. And as Omar and I were planning this session, a paper dropped into Nature with the first author of Barry, and I really thought, I wish I'd done that. So we contacted the authors, and the authors decided that Astrid should be the person to come and present this. So from Oxford, professor of virology and immunity, she's worked a lot on COVID and stuff like that. But has also done this serious bit of work, and I really wish I'd done that.
Thank you, Alasdair. It's a very multidisciplinary study, and we are 4 last authors covering a lot of, very different fields. So I'm going to take you back into the past. So we looked at the ancient origins of genetic risk for MS. And I'm just going to go very quickly through this. As you all know, it's a common, often disabling disease. It starts with an inflammatory phase, and we have peripheral immune cells that attack the CNS, leading to demyelination, and it progresses to a neurodegenerative phase. Neurons get transected and die, and we end up with irreversible disability. We have... sorry. I hope you heard some of what I said. Okay, thanks for the thumbs up. So we have talked about MS for about 150 years, but how prevalent MS was in the past, we don't really know at all. We do know that today MS is mostly seen in young women, but also in some young men. There are environmental risk factors that precipitate MS in susceptible individuals, and we have over 2.5 million patients. The risk factors for MS, well, we have about 30% genetic risk, but the rest is covered by environmental risk factors. Those can be very varied. Infections, nutrition seem to play a part, smoking, etc. And they can all trigger MS in genetically susceptible persons. MS prevalence varies greatly worldwide. And if we look in Europe, you can see a north-south gradient. Here are the number of MS cases. The prevalence is highest in Scandinavia and the British Isles and in countries where descendants of people from these areas migrated to. And MS prevalence has increased quite a lot during the last 50 years. Now, this north-south gradient suggests that it could be caused by environmental factors because temperature, humidity, sunlight changes from north to south at these latitudes quite a lot. But also we know from archaeological evidence that European populations are not all the same. I mean, you just have to look at us. There are differences in how tall we are, what hair colors we have, the predominant eye colors. So we are all mixtures of ancient populations. And we set out to answer the question where this genetic risk factor for MS originated. Was it in all ancient populations, or was it a specific population that had a great impact? Nobody really knew. And we also wanted to examine, to the extent possible, what was the possible triggers of this genetic risk. Was there any selection for certain gene variants, and what would be the drivers of this selection? So I'm going to take you way back. These two figures show similar things, but in a bit of a different way. So bear with me as I take you back to the first modern humans coming into Europe. The out of Africa population that entered Europe about 45,000 years ago. They very quickly split up in Northern Europeans and West Asians. And you see this here. Here you see the number of generations since the current time that has gone past. And here you see the estimated population sizes. Now, the Northern Europeans split up in Western hunter-gatherers and Eastern hunter-gatherers. And these two groups did not really mix for about 20,000 years. So they evolved quite separately. The West Asians split up in Anatolian farmers and Caucasus hunter-gatherers. So Caucasus hunter-gatherers lived in Caucasus and the Pontic steppe, which now covers Ukraine, parts of Western Russia, and parts of western Kazakhstan. Anatolia is pretty much what we now call Turkey. The Anatolians became farmers around 9,000 years ago. Farming was invented in the Fertile Crescent in the Middle East and then spread to Anatolia, and then over a couple of thousand years, northwards and westwards in Europe. On the Pontic steppe, people started to herd big flocks of cattle and sheep and goats and lived very closely with them. They were able to ride horses, and they invented wagons. And these people, I'm not quite sure where, there it is. These people mixed with eastern hunter-gatherers and generated a population we call the steppe people. And one of the big groups there were called the Yamnayas. Now, farming and pastoralism were very successful ways of living and created big population increases. And these increases led to migrations, especially of the pastoralists. Now, today, modern Europeans are all a mixture of these ancient populations. If you look at an Italian, there's a lot of farmer ancestry, originally from Anatolia, some western hunter-gatherer, but not a lot, and some steppe. If you look at a Scandinavian, here we have a Norwegian, there's a lot of steppe ancestry, some farmer, some western hunter-gatherer. And an Estonian would have even more western hunter-gatherer ancestry. We wanted to interrogate whether the risks were in any of these populations specifically or in all of them. So we used the result of GWAS studies for MS. Now, GWAS has uncovered about 200 non-HLA risk genes and 32 HLA-MS risk genes. So genetic risk to MS are often found in genes or next to genes that deals with our immune responses. And HLA genes confer the greatest risk, especially the HLA variant, HLA-DRB1 1501. But we do not have any real thorough mechanistic understanding of which role the immune system plays in MS. And that makes our targeting of the immune response somewhat imprecise. Now, to do this study, we used a lot of old samples. This shows the average age of the samples colored, and you can see the extent that we're covering and the number of samples. This is actually the largest ancient genomic data set that exists so far. And it covers the Stone Age, well, about 14,000 years ago to a couple of hundred years ago. And we quantified present-day Europeans' genetic ancestry using the UK Biobank and the results we got from the analysis of these ancient populations. And we determined whether and when MS risk genes had undergone selection. And we examined whether targets of selection were specific to any particular lifestyle, farming, pastoralism, hunter-gathering. And we also examined environmental conditions that may have caused selection for disease risk variants. So we found that MS risk was robustly associated with steppe population ancestry. Here you see the different ancient populations, and this is the risk ratio. And steppe ancestry was highly associated with risk, and particularly in the HLA region, and most strongly with the HLA class 2 region. The other ancestries actually seem to provide protection against MS. And we also examined this further by trying to see how robust our results were by assessing ancestral risk scores, both over the individual and using all SNPs, so that's the whole genome. And again, steppe ancestry was strongly associated with MS risk, the other ancestries weren't. Now, if we look at the strongest HLA variant, HLA-DRB1501, then this figure shows the x-axis is time, this is the present, and this is the frequency of HLA-DRB1501. And until the Yamnaya formation, the mixture of the Caucasus hunter-gatherers and the Eastern hunter-gatherers, we only found HLA-DRB1501 in one sample from Italy here. But during the Yamnaya formation, suddenly we found increasing numbers of samples with HLA-DRB1501. So it looked as if there was some selection going on here that favored the survival of those who carried this particular HLA variant. And this figure shows the frequency of HLA-DRB1501 in Europe today, and you can see a very strong north-south gradient. Now, we developed methods to test for selection on all SNPs. And this is a figure showing that selection. This is time. And here, this signifies that there is selection going on because the risk goes up. We always start with the highest risk variants and see what happens to them. Do they increase or decrease over time? And if you look at just HLA-DRB1501, we used 4 SNPs that people have published as markers for this HLA variant, and we found that it was strongly selected in Caucasus hunter-gatherers and a little bit in Eastern and Western hunter-gatherers, which goes well together with our overall analysis of the selection of these MS genetic risk SNPs. Because the Caucasus hunter-gatherers and the Eastern hunter-gatherers are those that created the steppe people. So the selection is driven by steppe ancestry. And there was a strong increase in MS risk gene frequencies about 5 to 2,000 years ago. And that risk increase coincided with the Yamnayas bringing risk genes into Europe. And the Bronze Age was a really turbulent time. It was a time where you had big population increases because of the technological advances in farming and in pastoralism. And those big population increases created a need to find new places to live, to migrate, and that resulted in clashes with the people who already lived where these steppe people migrated to. And I'd like to tell you a little bit more about them. They emerged, as I've told you, on the Pontic steppe and in Caucasus, and about 5,000 years ago, they started to migrate into Europe, particularly northwards and westwards, and also into Asia. These people were the first true nomads that entered Europe, and they had this enormous technological advance that were the ability to ride a horse. And they had invented wagons, and they had invented charts that could carry warriors, and they had a very extreme diet. They only ate meat and drank milk and consumed dairy products. When you look at the archaeological evidence, it looks as if they almost avoided eating grains. That was very much in contrast to the farmers, of course. And as a result, these Yamnayas were about 10 to 15 centimeters taller and more robust than Europe's original people. So when they arrived into Europe, they must have been a terrible sight. And there are numerous archaeological finds that show that the clashes between the original European people and the Yamnayas were often very violent. This poor guy has had an arrowhead through the cheek, and he was found with another in his sternum. And the Yamnayas left a very great genetic legacy, particularly in Northern and Western Europe, a legacy that is as great as the legacy of the Anatolian farmers in Southern Europe. When we saw this, we hypothesized that these novel lifestyles, farming and pastoralism, changed the complexity of the pathogen challenges that people were exposed to. And the increased population densities also increased the risk of transmission. So you have an increased risk of transmission of old pathogens, because there are more people living more closely together. And also you have an increased risk of being infected by zoonotic pathogens that can adapt to spread between humans also. These diseases are often very deadly, and there will have been a huge selective pressure that favored people with certain genetic variants that enabled them to survive these diseases. At this time, and until about 200 years ago, about 50% of all children who were born died before they were 15. So child mortality was extremely high. But today, some of these immune gene adaptations that in the past would have favored your survival might today increase your risk of autoimmune diseases, including MS. We therefore investigated what other effects the SNPs that were associated with MS risk had. And we found that most of the SNPs that were under positive selection also were associated with protective effects when we looked at different pathogens or different infectious diseases. And bear in mind that when we look at associations between the SNPs and the pathogens, we are only seeing the top of the iceberg, because we can only detect protective effects if these infections have been examined with blood. So we need to have good GWAS studies on them. We need to have good data on them. And many of the diseases that were very deadly, like smallpox, plague in the past, we have no GWAS for because they're not circulating anymore. And also, for many of the diseases we vaccinate against, we have either no GWAS or GWAS studies that are not very robust because the number of cases is quite small. So we are really only interrogating a small part of the whole pathogen challenge that people were exposed to. And many of the diseases that we found protection against were crowd diseases. So crowd diseases are diseases that only spread in large communities, like flu and measles, rubella, mumps. They were not really transmitted between small, isolated hunter-gatherer groups. So the result of our study kind of raises the question, if we have an unbalanced immune response today, because our immune response that we carry are the result of our ancestors' adaptations. And for thousands and thousands of years, our ancestors has been exposed both to bacteria and viruses, but also to parasite infections. Parasite infections are often long-term infections. Some of them can last for decades. Now, bacterial and virus infections triggers pro-inflammatory responses, but parasite infections triggers anti-inflammatory responses. It is important to remember that anti-inflammatory responses mute the effect of the pro-inflammatory responses. So if you need to have a certain level of pro-inflammatory responses to efficiently combat a pathogen, and you are at the same time infected with a parasite, then there will be a selection for very strong pro-inflammatory responses in order to reach an efficient level. But today, we have taken away the parasite infections. So we don't have that background of anti-inflammatory responses, but we still have the ability to generate really strong pro-inflammatory responses. And therefore, we might overshoot the pro-inflammatory responses to something that is more than that is necessary to combat the bacterial and viral infections that we are exposed to. And that might be something that's quite general in all of us, that we have a fairly high level of pro-inflammation. And in those who are susceptible to autoimmune diseases, including MS, sometimes this overshooting of inflammation might trigger these diseases. Now, there has been a few studies, small studies, on treating MS patients with parasites from pigs. And those studies have not been very successful. And the results have been very varied. But one of the things that come out of this is that actually, if you want to treat patients with parasites, you shouldn't use pig parasites, because the Yamnayas did not eat pigs. They had no contact with them. And I'm not suggesting that patients should be treated with parasites, but if we could identify some of the parasites, we might be able to identify certain epitopes that we could exploit to trigger anti-inflammatory responses in a safe way that might be rebalancing our immune system in a more gentle way than what we're currently doing to the immune system. Now, the conclusions of this story is that inherited MS risk is associated with step ancestry, and that many of these MS risk genes were brought into Europe only about 5,000 years ago. And that the MS risk genes have undergone positive selection because they were highly beneficial in the past. And our analysis suggests that this selection was likely driven by pathogenic challenges coinciding with these quite dramatic lifestyle changes. And the implications is both a demystification of why does something like MS exist, and the frequency that it does, and might also trigger new ideas for how you could go about thinking about treatment for MS and other autoimmune diseases. And also it demonstrates how the genetics and habits of all our ancestors really impact and shape our health today. And lastly, I would like to thank the main drivers of this study and also all the co-authors. There's a lot there because we both have samples, we have archaeologists, immunologists, neurologists. So this is, I think, a beautiful example of a multidisciplinary study and also a multinational study. From the UK, it was particularly Oxford, Cambridge, and Bristol, but there are many other universities all over the world who were involved in this. And the Lundbeck Foundation for funding the study, and you for listening. Thank you very much.
Thank you. As the microphone's making its way over there, there are therapeutic hookworms, and Chris Contanescu did a really nice study of hookworm infestation, which did show a positive effect in MS. It wasn't that well tolerated. Yeah, thank you.
Hi, fascinating story, and I wish I was doing this study too. I want to ask, from the beginning of the slides where you showed that there is this population with increased risk for MS, is there anything we can learn from the populations that do not have increased risk of MS? Is there anything we can learn in terms of protective factors there? And yeah, I forgot my second question, but this was the main one.
I don't think there was anything that they did that, but what seems, well, we know that DRB1501 is associated with protection in Europeans, not in Asians, with protection against tuberculosis, for example. And tuberculosis can be transmitted from cattle through milk to humans. So there might be something about the way they had really an extreme diet, being so dependent on milk. And of course, that was not pasteurized in the old days. So I think what we can learn is that lifestyles matter, and also that our immune system is adapted to a complexity of pathogenic challenges that we do not have today. But what we have today is perhaps an even greater assault of bacteria, and especially viruses, than in the past. So today, because we live so closely together and we do not really take many precautions to avoid getting infected, we are assaulted by all these pathogens all the time, and people are sick during the winters, and the children are sick all the time, and we are challenged in ways that we weren't in the past. And that might trigger a sort of continuous higher inflammatory state in all of us than what would be useful. And that might endanger us of being in a higher risk for a whole range of diseases. I mean, I cannot think of anything where a heightened inflammatory state is really beneficial unless you're actually infected. So, on the other hand, I can think of many diseases where inflammation plays a role, and it's not a good one. So maybe we could sort of use this to think about how to maybe prevent being infected all the time, in smart ways, like the current pandemic has shown us, that pathogens that are transmitted through air, you can actually prevent some transmission if you ventilate or you open a window or you have HEPA filters that filter the air.
Now Astrid, brace yourself.
Yeah.
Because Stephen Saucer has a question.
Oh.
So I wanted to ask you about Sardinia. So Sardinia has a very special place in the epidemiology of MS. It's
Yes.
probably the highest rate of MS anywhere in the world, and yet there's very little 1501 and no significant step ancestry there. So I wondered if you looked at Sardinia and thought about Sardinia.
We haven't looked at Sardinians particularly, but Sardinians, as far as I understand, have a very special genetic makeup relative to the rest of the Mediterranean. So they are a very special group of people, and it would be wonderful if we were able to interrogate
So there is a really
them.
nice paper that they did where they look specifically at the BAF mutation, which only occurs at any high frequency there, and that had clearly been subjected to malarial selection pressure, which was very common there. My second question was, how many people do you need to look at in order to estimate the allele frequency reliably? Because it's fantastically impressive to look at all these ancient DNAs. But if you want to judge the true allele frequency in that population that was thousands of years ago, how many people do you really need to look at to get a sort of realistic estimate? Isn't it possible that perhaps if you just had a few more Italians, you might have estimated a slightly higher
End of question.
frequency
End of
1501?
question. Brief answer, Astrid.
I mean, you always want more cases, right? You always want more samples, you always want more data. But this is the data that we had. And
So I think I know that Astrid and Steve will agree that you want more data. And on that perfect, harmonious point, I'm going to bring this session to an end. I want to thank Astrid for a fantastic talk. All session. Thank you very much.